I did want to say one thing about your homework that just really pointed out is, I think one of the questions about figure 2 is asking for, I ask for evidence of ER stress. And insulin signaling, okay? So you're going to have to define, you're going to have to decide what is ER stress. ER stress would be those events that are universal to all cell types, whether or not they are responding to insulin, okay? And insulin signaling is specific for insulin signaling, okay? So they aren't the same, okay? Because some cells under ER stress don't have faulty insulin signaling because they might not be responding to insulin. Okay, so make sure you make that distinction. And I think, is it the second question that asks you that?
Yeah.
Yeah. Okay. I just wanted you to point that out. Don't think those two events are one and the same. Okay. And I think. And then the other thing, just to remind you, even though I asked very specific questions, and I did on the Invokana paper homework, please write it as a paper. Don't say, here's the answer to question number 1, here's question number 2. Just write a paper. Make sure that your paragraphs contain one idea, not multiple ideas. Okay, so think about how you would organize a paragraph. Okay. I'm trying to get my picture bigger. Okay, so I think it's time we get out of the ER. Although, like I said, I could spend the rest of the semester in the ER because we have not hit all of the topics. So today we're going to start, actually we also end chapter 12 and start in on chapter 13, which is sort of a continuation of what we have been doing. But we're paying more attention now to how do you move from the ER to the Golgi. So far all we've done is get into the Golgi, and now we're thinking about, so how do you move to the Golgi? Why don't you get lost going, why don't you end up in the lysosome, or a peroxisome, or a nucleus, something like that. Okay, so we're going to take a look at, I'm going to give you the overview of what's going to be happening. And I'll tell you, we're only going to focus on one aspect of the vesicle transport, because if I cover one aspect, you can apply it to all the other places. So far we've seen, and we know now the difference between an integral membrane protein in the ER and a luminal protein. In the ER. Okay, and there's I don't think I've done this yet, but I'm going to put, yeah, I haven't, I just forgot. This article, which I really like, but it's way too much detail. I'm going to put this on Scholar, and instead of calling the proteins luminal proteins in the ER, they call them soluble proteins in the ER. In other words, they're just not integral membrane proteins. Okay? Some proteins that end up as integral membrane proteins in the ER membrane are actual ER resident proteins, and you should be making a list of those. Okay, so far you're making a list of different kinds of functions. Are they G proteins? Are they protein kinases? Now you should also be making a list of what are the ER resident proteins. Are they luminal or are they transmembrane proteins? So do make a list of those proteins that belong in specific places along the secretory pathway. Okay, and there are luminal proteins that reside in the ER and ought to stay in the ER. Okay, can you name one?
Yep.
BIP. How about another one?
Calnexin. Calnexin.
It helps fold lipoproteins.
Is it Calnexin?
Calreticulin.
Reticulin.
I know, I get the two mixed up. I
Okay.
get mixed up all the time. PDI, okay? And you could probably name some other proteins, okay? How about an integral membrane protein that resides in the ER?
Translocon?
Yes, the translocon. Anything else? It's not calreticulin, it's calnexin. Anything else? Well, maybe that's one great activity for you to do, is go through the proteins you've covered so far and decide, are they ER resident proteins, integral membrane proteins, are they cytoplasmic proteins? We probably will be hitting, we've hit some cytoplasmic proteins and like the proteasome, but we'll be hitting more, okay? So if the protein is not a member of the ER family of proteins, then it's going to move on to the Golgi apparatus. Everybody will move on to the Golgi apparatus. So really, there's only two fates for proteins that are properly folded, and that is they're either going to go on to the Golgi or they're going to stay in the ER. And just to remind you, these guys, if they're integral membrane proteins, at the C-terminus have the amino acid sequence KKXX, okay, for lysine, two positively charged amino acids, anything, at the C-terminus. Or they could have KXKXX, okay, so the lysines could be separated by another amino acid, okay? So we're gonna have to figure out, we'll have to look at later who reads that, who detects the KKXX, okay? And we've already described that these are KBL sequences. So resident ER proteins that are luminal or soluble have a KBEL. Okay. If they're properly folded, okay, these guys are going to need to bind to a cargo protein. Okay, in order to capture those proteins in a vesicle to move on to the Golgi, they need to be loaded onto a cargo protein. So that's just really to ensure that, for example, insulin, which should be secreted, is going to be able to get to the Golgi. So insulin is going to bind to a cargo protein that then ends up in a vesicle. There's no way it can float into the right kind of vesicle, just by diffusion, like, we don't accidentally pick up insulin and take it onto the Golgi. It's selected to move on to the Golgi. Okay. And these guys have address labels. The cargo proteins have address labels, and we'll take a look at a couple. Okay. Okay, so some of them have diaromatic amino acids, and I'll mention this again later. So at the C-terminus, you find, name it an aromatic amino acid. Phenylalanine, or tyrosine, or tryptophan. So we're going to look at a protein that has two phenylalanines at the C-terminus. Or they could have acidic amino acids, which means they're going to have either D is aspartic acid, E is glutamic acid. Both of them are negatively charged amino acids, and they're usually separated from one another by some other amino acid. Okay? there's more than one protein that can pick up the cargo. And actually, that's what we're gonna be talking about today is, well, how do you select cargo for packaging? Okay, and then, we can move on from here. Okay, if a protein accidentally, that should have belonged in the ER, accidentally escapes and ends up in the Golgi, there's a retrieval pathway. Okay, so we're going to, the pathway, the forward movement and the reverse movement involve different proteins selecting cargo, but the process is exactly the same for both forward movement and backwards movement. So we're going to look at the forward movement and take a look at, so how are the proteins different, what kind of cargo is selected if you're going to have to go back to the ER. Okay? I think that's all I wanted to say about that. Yeah. Okay, so let's take a look at we're going to look at your colored picture. I haven't used this picture before, so which has been good.
AA stands for amino acids, so.
Yeah, AA is amino acids, yeah, you got it. And, if you've not taken the biochem lab, or the biochem course, I really don't care if you can name the aromatic amino acids or the acidic amino acids. So that information, that specific information is really for people who have had biochemistry. And now you can put together some stuff you learned in biochemistry. This is why you have to know the amino acid structure. Okay, we'll try it again. Getting closer. Oh. That's fine. My computer went to sleep. There we go. Okay. Just to remind you that they're showing an endoplasmic reticulum that is continuous with the nuclear membrane because it is. The nuclear membrane and endoplasmic reticulum are pretty much one and the same organ. Okay. During the selection of cargo and moving cargo forward, a lot of vesicles are forming on the ER surface. Okay, and so that region where vesicles are forming is called ERES. I don't care that you do not need to know that. Okay, but you should know that there is one part of the ER where cargo is being selected. So vesicles are forming to either move proteins on to the next stage or return proteins that should not have been sent on to the next stage. There is a compartment, and this is not described in your textbook, but there is a compartment called Ergic. Okay, and Ergic is not like the endoplasmic reticulum. It's different from the endoplasmic reticulum. It's not like the Golgi. It's different from the Golgi. It's sort of an intermediate compartment, and things seem to be coming and going. So there are some people who think this is where a lot of the selection is going on, because you'll notice that forward-moving vesicles are going to go through the Ergic. Recovery vesicles, getting stuff out of the Golgi back to the ER, are going to go through the Ergic. So some people think the Ergic vesicle is where a lot of sorting is taking place. Okay. At the other end, so a protein who ends up in this part of the Golgi will have to visit every single Golgi stack. Okay, so notice that the Golgi, to me it looks like pancakes, a stack of pancakes. Okay, and a protein that ends up in the first stack, or the top stack, we call this top stack the cis stack. That protein's going to go through the medial stack, and there's usually more than one medial stack. There's multiple medial stacks, it's just that these guys wanted space to do other things. So they only show one medial stack. And then the trans stack. So this is, in the trans stack, we see a lot of proteins forming vesicle in order to take proteins to various destinations. Okay, so the cis stack is the receiving end of the Golgi. Okay, so we could call this the cis face of the Golgi. And then on the other end of the Golgi, the trans face, or the trans-Golgi network, you can call it whatever you want. We'll see it, I usually call it the trans-Golgi network. Vesicles are fusing, forming again, to take the proteins to their final destination, which could be an endosome. It could be going out to a secretory granule, takes things out to the cell membrane, usually in a regulated pathway. Okay, stuff coming in from the cells. So here we got some endosomes, things coming in from the cell, can sometimes end up in the Golgi, not in high quantities. Okay, so I was going to say that an early endosome is sort of like an immature lysosome. Okay. A late endosome is sort of like a toddler lysosome or maybe a teenage lysosome, okay? And the lysosome is the lysosome. And the difference between early endosomes, late, and lysosomes sometimes has to do with what are the population of proteins in that compartment. So some people have distinguished between early lysosomes and late lysosomes by what do you find in that compartment. But the real difference is pH. Okay, so early endosomes are sort of acidic, and late endosomes are more acidic, and lysosomes are very acidic relative to the cytoplasm. So I don't really care if you distinguish between early endosomes and late endosomes. You can just call them endosomes. And we will, when we talk about endocytosis, we'll talk about recycling compartments, or the recycling endosomes. That's the first place I've seen it called recycling endosomes, usually recycling compartment. Okay? Let's see if I can move this over a little bit for you. Okay, so the other thing you'll notice is that there's some color-coded things kind of coating the membrane. Okay? These are different proteins that help form the vesicle. And there are three different ones. They're COP1, COP2, that are extremely similar to one another. And clathrin, way between the Golgi and the lysosome and the plasma membrane. So between Golgi, lysosome, and plasma membrane, you find clathrin. And it's also used in, clathrin is used in forming vesicles. We'll talk a little bit about clathrin. If we don't get it taken care of this unit, we'll probably start next unit with a little discussion on clathrin. Okay? Because COP1 and COP2 are really similar in how they work, not necessarily structure, but in how they work, I'm going to talk about COP2. Okay? So we're going to be talking about how do proteins that need to go onto the Golgi get selected and get packaged into a vesicle and move on. Okay? The other handout that I gave you shows you the difference in pH from one compartment to the other. And I think that's kind of important because we're going to see interactions being made at acidic pHs. Probably most of the interactions are going to be broken at acidic pHs. We're going to see that certain peptidases, enzymes that cut proteins, are going to be active at acidic pHs, but not at neutral pHs. So insulin, for example, needs to be proteolytically processed after it gets a signal sequence removed. It needs to be cut yet again, but it's not going to get cut yet again until it makes its way all the way to the trans-Golgi and the environment is really acidic. The protease that travels with insulin is not functional until the acidity reaches about 6, the same pH as the trans-Golgi network. So there's a lot. So that gradation of pH controls a lot of different processes. And that's why I just throw out, you do not need to memorize the process. I mean, I can't, you know, if you want to put it on your cheat sheet, then I'm perfectly fine with that. If you want to say that the ER, it has a pH of 7.2, but the cis-Golgi has, what is it, 6.7? Yeah, that's, what do you suppose is causing that? What is a transmembrane protein that has to be traveling along the way? Probably a proton pump that is active fairly early in the secretory process and continues to act all the way through the Golgi and onto the lysosomes. It may very well be the protein that ends up in the lysosome. I'm not exactly sure. Okay, so you got 2 pieces of information there. I like that. The colored picture came from this article I'm going to put on Scholar. It's the 2nd page. Another good picture that you can look at, I think is really good, is this figure. So when you get that article, take a look at this figure. This is a generic figure that describes both COP1 coat formation and COP2 coat formation. So there's generic enough to look, all you're doing is looking at generic ingredients, but it's still really good. If you can name each of the proteins for the COP2 coated vesicles, that's great. I really do like this picture. So that's why I'm putting this on Scholar. The in detail of information in the text is a little bit more than you need to know. But that's okay. Okay, so I am done with this. I'm going to turn it on. Oh well.
There's a rag on that part right there.
Looks like you're staring at me. Okay, good. Okay, so there's probably 2 major questions you need to be asking yourself as we go through this whole idea of vesicle transport from one organelle to the other. And one is, how are proteins selected? Okay, because this isn't a random process. This is a real tight selection process, even though there are accidental escapees. Okay, and then the other question is, and this is, I think, a tougher answer, is, well, once the vesicle forms, how does it know where to go? So when that vesicle buds off the ER, how does it know to go to a Ergic or directly to a Golgi? how does it not end up on, at some other organelle? Okay, so those are the two key questions. How do you select the cargo, and how do you tell the vesicle to go to a particular location? Okay? Mm-hmm.
I thought all the vesicles have to go through the urgic to get to the Golgi.
I think you got some that can, yeah. But notice that you got some that can go from the Golgi all the way back to the ER without going through the urgic. Yeah, so in this model, everything goes through the urgic as it's moving forward. Like I said, urgic is not even mentioned in your textbook. So, and this comes from, this is a model that comes from 2010. Your book is probably fairly generic and it tries to cover tons of organisms.
Should we work with this assumption that?
I would. You know, so if you're gonna go on a model, go on that model. Is that okay with everybody? Okay, so your colored picture becomes the model, okay? And so, yeah, thanks to Shelley for asking that. Okay. Okay, so there's a couple steps, or there's actually 4 steps involved, and then there are separate proteins involved in accomplishing each of the steps. You gotta select the cargo. And then you form the vesicle. Okay. Okay, so this is what's happening on the exiting face, or the exiting organelle. Okay, then you have to locate, how do I want to say that? The vesicle has to find the target organelle. And the vesicle has to fuse. With the target organelle. Okay, today we'll probably just get through the first 2 steps, selecting the cargo and forming the vesicle. Okay, proteins involved. Okay, it could be, is that too dark? I'm gonna get yellow. I'm worried that it's not showing up on the video. Cop 1 and cop 2. These are considered the coats that form around the vesicles. They have some other kinds of functions besides just being a coat, but this is considered the coat that forms around the target vesicle. Okay, and clap. Okay, I'm going to add some more proteins. COP1 and COP2, those coat proteins can select cargo, so they have binding sites to capture cargo. Clatrin has no capacity to select cargo. It just knows how to form a cage and create a vesicle. So clatrin uses adapter proteins. To select cargo. Okay, so the cargo that needs to be selected has to bind to adapter proteins. Okay, all of these are their formation and their dissolution, because you're gonna have to throw off the coat proteins in order to fuse the vesicle to the target organelle, and you have to regulate the formation of the coat in the first place. So the regulation of COP1 formation and COP dissolution, just uncoating or uncoating this vesicle, is controlled by ARF. Okay, you are going to see another protein called ARF in the next unit, and ignore it, they're not related. Okay, so you'll learn ARF in this context, but there's another ARF that has a totally different function. And SAR, and I can't remember. Oh, and Klassen uses ARF. Okay, and we're gonna take a look at COP2 and SAR and how those interactions are controlled. You can apply exactly the same analogy to understanding how COP1 and ARF work. Okay, to find to locate the target organelle and identify it, there are two proteins involved, two kinds of proteins. One is there has to be a protein on the vesicle that says this is what kind I am. Okay? This is sort of like also like a probe, but it is identifier. I'm a vesicle carrying such and such. But it is also used as a probe to find the target organelle. And the target organelle contains a T snare. So T stands for target, V stands for vesicle. The two find each other and they interact with one another in a way that's so tight it takes ATP to break their interaction. So a V snare is going to bind very tightly to a particular T snare. Okay? If the organelle, if the vesicle bumps into the wrong organelle, the V snare T snare combo can't form. Okay? So that takes care of, that's how you find that's kind of the landing point. There are also some other hints. Okay. RAB is sort of involved in moving that vesicle to a final destination. So RAB is also involved in finding the organelle or making sure the vesicle gets to the organelle. RABs, there's tons of different RABs. They have way unrelated functions. It's just like a group of oddball proteins. Another protein called NSF, and another protein called SNAP. Okay, so today we'll talk about COP2 and SAR, and then next time we'll talk about V snares and T snares. And in that picture that I thought would be worthwhile looking at, you've got V snares and T snares already in place. Okay? Okay, so